Okay, so today's video is focusing on DNA replication. The whole point of DNA replication is making a copy of itself. So the first question comes out to, well, why? Why is this a necessary thing? Well, if you recall, during the cell cycle, DNA is found in the nucleus. And the cell spends most of its life in interphase. And interphase has three phases. The gap 1 phase, basically we're doubling up on the amount of organelles, we see growth. And then primarily our focus is the S phase, and that's synthesis. So what is happening here is the cell gets so big that it no longer is efficient to move things around. The surface area to volume ratio is thrown off, so it leads to mitosis, cell division. So we need to make sure that we have a copy of DNA for both cells as a result. So the reason that we have DNA replication is to make sure that both cells have the same amount. And we end up with two genetically identical daughter cells when we talk about mitosis. Now, in order to make this happen, we have a couple players. And those players, notice how they all end in ASE. Remember, these are enzymes. So there's three major enzymes that we're going to focus on, but we'll also talk about a fourth that doesn't really get talked about as much, and understanding how one DNA molecule becomes two. So, very simply, what is DNA replication? So DNA replication is, I'm just going to make it as simple as possible, one DNA becoming two DNA. In essence, we are make a copy. So why does it happen? So the big thing here is cell gets too big, which means it needs to divide. And we need to make sure that both daughter cells have the same amount of DNA. DNA is found in the nucleus. And then when does it happen? This is happening specifically in the S phase of interphase. So kind of that middle phase within interphase. So we're going to talk about three major enzymes, and we'll add a fourth one in there just to make things a little bit more complete. So the first thing that we are going to talk about is what's called DNA helicase. H. Now what DNA helicase does is it unzips. And how it unzips is it breaks the hydrogen bonds. So what we see right here is our two strands, and this is going to be helicase right here. Notice how it broke the bonds between those paired bases. So we are separating the strands, almost making a fork. Step 2 is we have something called DNA polymerase. DNA polymerase is the builder of new strands. Now there's a little kicker here. Remember that DNA is anti-parallel, which means that we have opposite strands. 3' to 5', 5' to 3'. Now, DNA polymerase is only building in one particular direction. It is only building in the 5 to 3 direction of that new strand. So when we look at, for instance, this one over here, this new strand down here, put a different color, is going to be 5', and this new strand over here is 3', so 5 to 3, and then on the other side, 5 to 3 going the other way. So polymerase wants to work in this direction with this new strand to be built. And what's going to happen is it'll move around and simply add the correct bases and make that a new strand as it works in. When it is working toward where helicase is, and we call this the replication fork, when it is working toward the replication fork, it is a very continuous action. So whenever we are working in the 5 to 3 direction toward the fork, we call this the leading strand. It's very quick, very fast, makes things happen. Leading strand. Now, the other strand doesn't really operate as much, as continuous. We still have DNA polymerase is adding in nucleotides. However, it is moving away. So notice how we see this is moving away in chunks. Polymerase can't really operate as well going away from the replication fork. So what ends up happening is it works in little chunks. It'll have a chunk right here. It'll have a chunk right here. And this causes kind of these little gaps and these little islands. Now these islands that we see, these little segments that we see right here, those are called Okazaki fragments. And then obviously, we want to make sure that we have a continuous strand. So our last enzyme is what we call ligase. And this is the gluer of the Okazaki fragments. So ligase will come in and it'll take some of these free nucleotides that are floating around that we see right here, and it will add them to the appropriate spot and kind of fill in the gaps. It is the gluer to kind of make this a whole strand. So that's ligase. Now this strand, since it does take longer, it does have an extra step, whenever we are going the 5 to 3 direction away from where helicase is, we call this the lagging strand. There's simply just an extra step in regard to what is happening. So leading strand is a nice continuous motion, continuous build. Lagging strand, we would say, is discontinuous. This is what makes the Okazaki fragments. In which case we need ligase to come in to glue everything together. Now if you recall from our first slide, I said we had 4. The only one we didn't talk about here, helicase is the unzipper. Polymerase is the builder. It'll also proofread and make sure there's no errors. Ligase is the gluer. Only one you don't really hear too much is primase. And all I'm going to say with primase is primase kind of happens right here. It sets primers. And when I say it sets primers, it basically sets the starting points of both strands. So in this case, there might be a primer right here, which tells DNA polymerase this is where you begin. On the lagging strand, there may be a primer right here, there may be a primer right here, which leads to the gaps, which leads to the Okazaki fragments, those gaps that need to be glued down and solidified by ligase. It's just more of an FYI. The final thing is DNA goes through what's called semi-conservative replication. What this means, if this is our original DNA molecule, whenever it splits, both resulting DNAs that are formed still have one original strand and one new strand. So one original strand and one new strand. So semi-conservative, the reason why this is important is this protects against errors. So if you're always making a copy from the original, you're going to have less likely mistakes happening in the long run. If you were ever to take photocopies of things, make copies of copies of copies, you'll find that the ink is no longer crisp when you hit 20, 30, 100 copies. But if you're always making a copy from the original, you're always going to have the best possible outcome for what you're looking for. Okay, so that marks the end of DNA replication. If you have any questions, let me know. Thanks for watching.